We have one more talk in this session. It's going to be from Alan Cohen, who's a professor in the School of Public Health. And he's going to tell us about some novel proteomic methods to analyze not our stress test protocol for MiSBIE, but instead the awakening response, which is in itself a sort of stress. And I'm sure Alan will tell us more about that. So take it away, Alan.
Yes. Thanks so much for the invitation. Thank you, Martin, Caroline, Vanessa, for putting together this amazing symposium. Really happy to be here. The work I'm going to present today is work where Mole Liu and I were collaborating as senior authors. Mole was in biostatistics here, but he's now at Beijing University. And then Jiaying Zhao and Bowen Xu are the main students working on this project, and then several other co-authors as well. So this is really a joint effort here. And as Evan mentioned, this is the one talk in this session where we're not actually using that same stress course that you've seen, but it is still, I would argue, about stress reactivity. It's the awakening response. And I got stuck in an airport last night. I had trouble waking up this morning. We could consider that awakening as a stress, but whether we think of it as stressful or not, it's an adjustment the body needs to go through. And the body goes through many regulated adjustments, including the psychosocial one we've been looking at. So I don't think there's any qualitative difference between looking at how the body adjusts to awakening and how it would adjust to different other sorts of different things. It's an open question whether they're parallel adjustments or not, but it's a similar type of adjustment. So that's what we'll be looking at today. The broader context of what we want to get at, so Martin and I are the co-directors of Columbia's program on the science of health. We would like to define health scientifically. And so we start doing that by separating out what we call intrinsic health from realized health. Realized health is all the things you want to be and do and succeed at in the world. What's what matters to you. And so this could be different for each of us. It's different depending on our culture. Intrinsic health is the basic biology, which is unlikely to differ very much among each of us. We're going to have similar kind of homeostatic mechanisms that keep our bodies functioning well. And so here we're focusing on the intrinsic health chunk of it. And so we believe that intrinsic health emerges from the interactions of energy networks, communication networks, and structure in our bodies, and that this is a little bit parallel or analogous to a magnetic field. We had a great question on electromagnetic fields earlier. Here we're more using it as a metaphor, even though there are actual electromagnetic fields involved, but we think that the same way that you can't measure the magnetic field by looking at its components, you can't measure intrinsic health by looking at its components. You have to measure how that emerges and manifests at the organism level. And so we want to figure out ways to look at how the dynamics of the whole organism can be harnessed to figure out ways to measure this. And that's what we're starting to do. This is actually the very first study published towards that effort. It's a small pilot study. We've only got 12 individuals for whom we have data. But I think it's a good first step in that direction. This just came out a month or so ago. And so the more specific question here then is health status encoded in the dynamics of how the proteome responds to a stressor? So we're going to take the whole proteome. We got about 3,000 different proteins that we're measuring. Forgot to put it on here, but it's the O-link panel of about 3,000 proteins. And we're looking at right when you wake up, 30 minutes after you wake up, 45 minutes after you wake up, and then again in the evening. It's in saliva. And then we have, for each individual, 2 days worth of data. There were samples collected for 3 days, but we only had the funding to do 2 days for 6 individuals with MitoD status and 6 control individuals. And what we really wanted to do here methodologically was to figure out, like, there's thousands of proteins. There's different time points. There's different ways you can measure change between this time point and that point. There's just so many options for how you would analyze this data. So how can we construct a computational method or a statistical method that's going to get at what's actually happening with that proteome? And it's a lot of trial and error to see, can you figure out things that work there? But we wanted them to be unsupervised methods. And the reason we want it to be unsupervised methods is because supervised, what that means is you're going to say, okay, we know these guys are MitoD, these guys are control. Can we predict that from thousands of proteins over many time points? That's actually going to be pretty easy. We don't have a really big enough sample size to do it, but there's so much information in the proteome that would be trivial to say that we can distinguish people if we're trying to do that. The more interesting question is, when you look at the variation that kind of naturally pops out at you from those proteome dynamics, is that already giving you information on who's in the MitoD group and who's in the control group? And so that's what we wanted to see here. We wanted to develop unsupervised methods. If we're kind of blind to who's mito and mito-D in control, are we still going to see that difference pop out of our data? And so we came up with 3 different pipelines to do that. So as I've already explained here, we have 6 healthy individuals, 6 mito-D individuals, 4 time points per day over 2 days. And then the one element I haven't told you about yet that we wanted to compare is we wanted to see when we use the whole proteome, is that going to do better than if we pick out our favorite molecules? That is, maybe I can take the whole proteome and, okay, I get the signal, but it's really just due to the 2 or 3 best ones that have a lot of signal in there. So we also wanted to look at that. So that's this low D, high D distinction here. The low D would be picking out a couple molecules we think are going to perform well. IL-6 and GDF15 in general were kind of our go-to candidates there. And then the 3 pipelines we've developed, we're going to call them delta clustering, proteome elasticity, and tensor decomposition. The delta clustering, what we're looking at here is between any 2 time points, let's say between 45 minutes and PM, as you see in the graph there, things could change. It might go up, right? And when one protein goes up, does another go up with it? Is there a correlation that they go up together? And so we're going to look at that for every pair of proteins. So we get a 3,000 by 3,000 matrix, 9 million correlations, and about half that. And then we see if that we see if there's a structure there that is different between mito-D individuals and control individuals. Is that whole structure of how the whole thing changes different? The proteome elasticity, we're adding in one more layer of change here. Elasticity is a concept which you can't do with 2 time points. You need a 3rd time point. What we're asking with elasticity is, can you change and come back, right? You might be up and back or down and back, but do you get back, right? And so I'll tell you in a minute more how we do that. And then we're going to do that for each of these 3,000 proteins, and then use a method called t-SNE to see if we group, like if there's grouping differences between the mito-D and the control individuals. And the last one is called tensor decomposition, and I've got another slide on it later, and it's complicated, so I'm not going to get into it yet. So for the delta clustering results, here's what we found. In the top left, you're going to see this control panel. That's the 6 control individuals where we took their 3,000 proteins and looked at the correlation structure, and then we used hierarchical clustering to order those proteins so that it's in the right order. So you see these dark blue squares pop out along the diagonal. That's because those are sets of proteins that are tightly correlated together, right? And so then if you look at the top top right, you're going to see the 6 mito-D individuals. That's the other 6. And so what you see there is that there's no correspondence at all between the structure for the control and the mito-D. I should mention that the order of the proteins for the mito-D square is the same as the order for the control square. So we took the order from the controls and said, when we put them in that same order, do we see the same blue squares pop out? If we did, that would mean we're seeing the same correlations. We're not. So what we're seeing here is a very different set of how everything correlates. What's correlating with what as they change together? Quite different between the control and the mito-D. But maybe that's just chance. So let's take 6 random individuals, some mito-D, some not, and do the same thing, and then look at the other 6. And there, well, we've got a small sample size. We're estimating these correlations with a lot of error, so it's not perfect, but you can certainly see the same structures popping out on the bottom right as on the bottom left. And what that means is that if you take some random individuals, you are getting at something that is kind of shared across individuals. We have a big enough sample size, even with this small sample, to start to look at what that correlation structure looks like. It's just that it's different between the control and the mito-D groups. And then just to be sure we hadn't kind of picked the right random 6 and so forth, we did that for every combination of 6 and 6. And then we asked the statistical question, is the control versus mito-D group the most significant or one of the most significant ways you could distinguish it? And that's at 0.0411. It's because there's kind of a limited number of combinations, it always comes out at exactly the same p-value, but it is significant that there is this distinction that is the mito-D and the control are more distinct than any of the random groups. Then, as I mentioned, we want to compare that with GDF15 and IL-6. So when we do the same exercise with just those 2, any way we do it, we could never get anything that was close to significant with GDF15 and IL-6 to try to distinguish those dynamics. So that's the delta clustering.
Moving on to elasticity here. We were inspired by this article by Joe et al. In Cell Metabolism in 2023. So these are just a figure from their paper, and then like the definitions that we're using of elasticity here. So you can see that they were comparing young and old mice, but you can see that for the different gene expression levels they were looking at, you could see this kind of dynamic of elasticity. It goes up or down and then comes back. And so the three things we're putting together for the elasticity are the sign, so it's up or down, the amplitude, so how high did it go, and then the ratio of the two, did it come all the way back? And so if you have a very high response, but you only come a little bit back, you don't have much elasticity. If you only go up a little bit, but you come all the way back, you still don't have much elasticity. To get your elasticity high, you need to move way far away and come all the way back. So if we start off with our IL-6 and GDF15, we see that with IL-6, IL-6 didn't have back up one step. We're now looking at 3 time points. Those 3 time points are 0, 30, and PM. So we're skipping the 45-minute time point here. When we look at those dynamics, IL-6 actually doesn't even really show that dynamic in general. So it didn't really work for IL-6. GDF15 does, and we see that with the mito-D individuals, there is a higher elasticity of GDF15 than in the control individuals. But overall, we found that those were not the 2 best proteins to look at this for. So we chose the 2 best. If we wanted to get the 2 best out of the 3,000, they were 2 proteins I'd never heard of, and it's so small here I can't read it, but I think it's LSM1 and MEGF9 maybe? Anyway, those were the 2 best. And you can see that they don't distinguish mito-D from control individuals at all. The red is mito-D, the green is control. That's the middle panel. But when we look at the elasticities of all 3,000 proteins together, and then we run the t-SNE on it to kind of group it along the main axes of variation there, well, now we get a very clear separation between our 6 mito-D individuals and our 6 control individuals. So this dynamics of like, how is the whole proteome being elastic? Well, we're clearly seeing that that is different between mito-D and control. The tensor decomposition, I'm looking at the time here, I'm going to go through this. It's a difficult concept, and I'm going to go through it kind of quickly. So we may have to come back for questions. Roughly speaking, we can think of our data set as being organized along different axes. There's time, because we've got 4 time points. There's individual, we have 12 individuals, and there's proteins. We've got 3,000 proteins. And what we want to do is to kind of separate out, there's this kind of like 3 axes on which we can run something principal components like, and that way we can look at part of that variation in relation to another part. What does that mean here? Well, when we look at the time part, the first factor for the time part, you can see that the loadings for those 4 time points are 0.75, 0.44, 0.39, 0.31. They're all positive. So what that's telling us is, for example, like some proteins are high when other proteins are high. How high are the proteins? That's not what we're really interested in here. But if you look at the second factor, now we're seeing that baseline at 0 is high in relation to negative coefficients for 30, 45, and PM. So this is now a dynamic change factor, right? So this second factor here is of interest if we want to look at the dynamics of things. So we pulled out that second factor, and then we kind of basically ran principal components with respect to that factor. So what are the proteins that change with respect to that second factor there? And then with that, we can look at across those, the main principal components, are we able to separate out the mito-D and the control individuals? On the far left, again, you have IL-6 and GDF15 with the same types of approaches, and we really don't get much at all. But when we take, there's 2 sets of 3 axes in panels B and C. There, the separation's not quite perfect, but we can mostly separate out the mito-D and the control individuals with those approaches. So overall, like this third method is, I would say, less convincing so far than the other 2. I think it's promising, but I would want to see a better confirmation than what I have here with a larger sample. I think it takes more data to really show that this third one is working. But the first 2, I think, worked quite well. And so we now have 3 separate approaches that allow us to see how mito-D individuals and control individuals differ inherently in rather kind of obvious ways in terms of how the whole proteome changes
as it responds to a stress, in this case, awakening. So that there's kind of something inherent in the dynamics of the proteome that is different between these two. It's detected through multiple pipelines. These three are just the three we came up with here. I think the idea is more to show the principle of how it can work than to say, these are the best pipelines. And then in all three cases, the dynamics of the whole proteome work better than choosing a few best proteins, which suggests that it's not one or two things. I mean, maybe we didn't choose the right ones, but we tried to. So we don't think that it's the case that you could pull out just a couple proteins and get something that works as well as what the proteome as an ensemble does. Doesn't mean you need all 3,000 proteins. We could probably drastically reduce that number because there's a lot of redundancy in what we're measuring, but it does mean that there's probably not a very tiny set that is containing all the signal. So the next steps would be to validate this in other data sets with other stressors and health distinctions, so not just MitoD versus control, but let's say frail versus non-frail older adults or things like that, to develop better pipelines. And then we really want to expand this kind of more generally into methods where we could detect signals of intrinsic health. So in terms of the implications for MitoD patients, I would say that we're on the way there, but we have more work to do. This is just the first step of a lot. But I do think in broader terms, we're starting to show the importance of looking at system dynamics and the whole system rather than just a few parts that we're isolating and trying to put together like Legos. We really need to look more comprehensively and holistically at how the whole system is functioning. And I don't know if there's time for questions or not, but I'll end there. Thanks.